Hi, everyone. I think we're going to get started. Sorry, I got sidetracked chatting with our speaker. It's my pleasure to introduce Dr. Benjamin Wong Blonder as our seminar speaker today. Dr. Blonder is currently the Hayden P. Reinecker Distinguished Professor in Forest Genetics, and he's an associate professor in the Department of Environmental Science, Policy, and Management at Berkeley. He did his PhD at the University of Arizona, after which he did a couple of years postdocing at Oxford. And then he came back to Arizona and spent 2 years as an assistant professor at ASU in the School of Life Sciences. And he still has some active collaborations in the state of Arizona, including the University of Arizona's Sky School, which is a nonprofit organization focused on providing inquiry-based outdoor science opportunities for K through 12 students. And so he co-founded that as a PhD student in 2012, and it continues to this day. I think Dr. Blonder would consider himself a plant ecologist, but his work is really creative and wide-ranging, and he uses a plethora of tools from mechanistic and trait-based modeling to answer some fundamental questions in community ecology and biodiversity. I first met Dr. Blonder as part of a Gordon conference on predictive ecology that was co-organized by Shannon Ledoux and attended by some of us here at Cary. And just from my brief interactions with him at that conference, it was clear that he's just a really creative and wide-ranging thinker that does a really beautiful job of conceptualizing complex questions and thinking about how to answer them in structured, robust ways using a bunch of different tools, bringing those tools to bear on these questions. So we're really excited to hear your talk today, Dr. Blonder, and looking forward to your engaging with our community at Cary. Thanks.
Well, thanks very much, Barbara, for that really kind introduction. This is my first time up here at Cary, and it's really nice to get to see where you all work and all the great science that's happening here. So I'll be talking today about a project which is actually in none of the areas that Barbara talked about. This is sort of a new pivot for me. And this is work that's just coming out in a journal in a couple of weeks, so I'd be really glad to have some feedback and see what you all think about some of these ideas. The main focus of the work is on community assembly, one of these classic topics in ecology, and I thought I would begin by illustrating the challenges of it by showing you a couple of pictures. So this first picture, right, is a farm or maybe somebody's garden, and I think many of us have tried to plant a garden, which is effectively a community assembly process, and we put some seeds in the ground and we hope to harvest something at the end, but it doesn't always come out that way. So even in this very simple controlled system, predicting the future of a community, that is these plants, can be very difficult. Then as a second illustration of the challenges, I just want to show you a nice little time series from a field project that I have out on the Rockies. I'm going to be showing you some maps that look like this. These are vegetation plots, and the different circles are different individual plants, and then the colors are different species. And we have a lot of these plots, and the details don't matter, but if I just advance us through 11 years of data, you can see a lot of stuff happens in the system. And I personally find it very difficult to explain why the things that have happened actually did happen the way they did. So these two cases, a simple hands-on one and then a longer-term data set, I think both illustrate the challenges of community assembly. And I'll offer in this talk some ideas on how we can do better at getting at these questions. So the outline then will be first to just give you a little bit of conceptual foundation to think about some of the issues at stake. Then I'll talk about a particular kind of problem in the area of community assembly we might like to solve. And then we'll try and solve it through something called love. We'll see how well it works, and then we'll consider more what it would look like to take these ideas, if we like them, and bring them into the real world. So first, the conceptual overview. I would say that if you look in your community ecology textbooks, or just an ecology textbook broadly, you get a very mechanism-first idea of what the field is. When we think about species coming together in communities, we think about the processes that bring them together. So for example, how important is chance? How important is competition? How important is predation? What kinds of predation exist? These are the things that our undergraduate educations give us a lot of detail on. And then when we scale up to larger communities, we start to ask questions about, do these species coexist with each other? Is it a stable coexistence? How do populations change in order to maintain or not maintain that coexistence? Peter Chesson, of course, has done a lot of work in this area. And then more recently, people like Mark Velland have tried to put together all of these ideas into these conceptual syntheses. But they're all very focused on the processes. More recently, I think there's been a sort of push to say, well, whatever the processes are, can we predict anything about the future in a community? And some of the pioneering work on this was done by Paul Ketty, where he talked about assembly and response rules in the early 90s. And then more recently, as the world of ecological forecasting has grown, there's been a number of calls in various areas, especially in applied ecology, to move towards what Jeff Houlihan and others have called the priority of prediction. So there's this tension in the field right now between these two different angles. And for some people, the prediction side is maybe too hard and not worth going towards. Dan Simberloff in the early 2000s had this very influential publications where he had a broadside against the effort of prediction, right? Where he asked, is it just time to move on? Is it too hard? Is there too much contingency? Is there too much complexity? Should we just give up on some of these prediction goals entirely? And of course, it's a straw man of a sort, but it underscores, I think, some of the challenges in making progress in these areas, given the vast biological complexity we're all familiar with outside. Yet at the same time, we have many people who feel very strongly that if we could do a better job in these areas with prediction in community ecology and community assembly, we could solve a lot of important problems. And you'll see certainly in recent literature, this is 2015, articles, I think, speaking more towards the health and the medical world, arguing that community ecology does have a very large role in health challenges. And I know there's a lot of disease people in the room here, so this is not news, but there are these tensions and these different threads trying to bring all of these different ideas together. So it would be great if we could make some more progress here. There are, of course, other applications, applied community ecology applications that are linked to community assembly as well, and I'll just give you a couple illustrations here. One, for example, might be questions of revegetation and restoration. So suppose you have a mine site or a landfill, you'd like to revegetate it. What are the species you should put out that are going to best revegetate that site, that are going to persist in the future, that are going to coexist with each other, that will do okay in the future, that can tolerate the metals, whatever. Another question might be if we're trying to produce certain biomolecules at scale in factory settings, maybe things like fake meats, but many other chemicals as well. How could we produce these metabolites? What microbes are we going to use? How are we going to put them together? How are they? What resources do they need? Can they keep on producing? How do we keep the successional stage where we want it to be? So again, community assembly. Then a third question, lots of infectious diseases. Maybe you get some sort of parasite or other challenge in your gut microbial community. Can you cure that? Here we have an example of a fecal transplant, which is effectively a community assembly solution where you disperse one community into another location. It seems to work for some diseases in some cases, but not others. But we don't really understand why. So there's a lot of exciting opportunities there. And then we also have challenges of invasive species management, like in this desert case, where you see a Sahara mustard, this let me use the pointer. This large species here in the bottom left, an invasive species in the Mojave Desert, and you might like to get rid of it. So what are the actions you could take to shift that community from one state to another? So I show you these just to illustrate some of the challenges at hand. So with that in mind, let's talk about some ways we could make progress. I'm going to argue that there are two basic problems that are interesting to solve in this space. The first is simply prediction. So if you start with a community in one state, here are individuals of different species illustrated as symbols, and you take some action, or you don't take an action and you just wait, this community might shift into any number of future states. Which one is it going to be? Could you predict that? So this encompasses questions like, will certain species coexist? Or will an invader establish? Or will biodiversity go up or down? Those are all prediction problems. Then the second set of problems, which are more on the applied side, are prioritization questions. So this is a set of questions where we might like to ask, what is the best action we could take to shift a community from an initial state to a desired state? So if you think about all the number of things you could do, you could add a species, you could delete a species, you could change the environment, you could move some people, you could ask people to behave differently, any number of things. One of those things might do better than others at achieving some goal that you have in mind. So which action should you pick? And this might get you at questions like trying to maximize biomass or functioning, or increase biodiversity, or make something invasion resistant. But you would need to know how to pick that action. So there are lots of approaches to prediction and prioritization that I would argue sort of spread out along two axes. How much do you want to know about how the system works? and then how complicated actually is the underlying system. So over on the bottom right, you don't really need any theory. If you want to know a lot, but it's not very complicated, you just look at it, you do a couple experiments, you know, and you're quite happy. Then sort of along the main diagonal here, there are a range of different approaches that ecologists have come up with, from relatively simple ones like species distribution models that you stack on top of each other, where you don't really know why the species are there, but you have a lot of data. You don't really account for species interactions, but it might work well enough for some cases. And then you have some very complicated approaches like finding a dynamical model and then parameterizing it, getting all the species interactions, complicated system, complicated work. Maybe it works in some cases, and then a range of other sorts of things that fall in between. And if you go to the very top here, there are some cases where maybe you don't understand too much from the Western ecological perspective, but the system's complicated, but you can still learn something. And these would be cases where I'd argue that either expert or traditional knowledge get us actually relatively far in these cases. And if we keep going up in complexity, I would say the other place we get to is just give up, right? It's too hard. This is the Dan Simberloff theory of community ecology. So love, which is what I'm going to be talking about in a second, and I'll define it as well, is something that sits sort of on the top left of this diagram. It's cases where we don't actually want to know that much about the system, but the system's really complicated. And you can see that this sort of sits in a space where there's not many alternative options. So that's what we'll be talking about today to get at these problems. Okay, so now let's talk about what love is. And I will define it in one or two more slides, but I'm going to leave the acronym out for just a second. So when would you want to do something like this? As we talked about, we're up in this part of the diagram. So this would be a case where you don't know much about the system. You don't know much about the mechanisms. You don't know which dynamical models would work. You don't have any parameter estimates. And if you wanted to get data, the data are going to be very hard and difficult to obtain. So this is kind of a lot of ecological cases, I would argue, where you don't know much, but you still need to make some progress. And maybe there are also cases where being able to do the prediction or the prioritization is more important to you than understanding how the system works. If you can solve the applied problems, you're happy. If you want to know about mechanism, you're unhappy, but that's okay. And then also cases where the system is complex enough that it's worth taking a new and different approach to any of the other ones that we've seen before, where these other approaches are going to fall down. So that's the problem space that we're working in. So how would we do this? Well, we would use this approach of learning outcomes via experiments. That's where the love part comes in. And the idea here is actually pretty simple. You experimentally do something to a community. We'll talk about what that something could be later, but you do something. You start it in one state, and then you wait a while, and you observe what happens to the community afterwards. Then you repeat that process several times. And hopefully by basically pushing the system in enough directions, you can learn what else it would do if it was pushed in new ways. The kind of data that you need for this is similar to the sort of data that are carried out in biodiversity ecosystem functioning experiments. So here's the Cedar Creek Altur site, and you can see these are experimentally sowed communities, and they look different, so they've had different outcomes over the decade or two that they've been running. But this is the idea. You could also do this as microbial inoculations. You could do this as, pick your system, but as long as you're manipulating the system in some sort of way. So let's talk through how this would work and why it might be a useful approach in a little bit more detail. The kind of data that you would put together to make this work would be, first, you would gather some information on the initial state of the community and the initial environment that we find ourselves in. In this case, I have this illustrated just as empty. So this is sort of like a community with nothing in it, and the environment, we'll just say, is at a baseline level. Then we take some sort of actions. And here I have 4 actions illustrated. The top, I add 5 species. The next one, I add 3 species, et cetera, et cetera. After I've done that, I wait a while. And then I observe what the abundances of those species is after some period of time. And here you can see in the top case, the blue one seems to do well, the green one does too. In the third case down here, the orange species excludes the green species, and in the bottom case here, 2 of these species exclude the red species. And then we can also, with these data, go and look at them and just with our own human opinions say, well, which ones do we like? So we can score them. Maybe this one is very palatable to us, this one is very unpalatable, et cetera. So we can put our own values on top of these as well. In the language of prediction then, If we had data like this and we knew what to make of it, we could then, say, take some other combination of actions and initial states and then try and predict the abundance outcome. That's what we'd like to try and be able to do. And in prioritization, we would say, we want a really big desirability. What sort of outcome would we need? And then, therefore, what action should we have taken to get it? So we're going backwards. So to be able to do this, we're going to take a mechanism-free approach. And the workflow here is to first figure out what we're trying to achieve, then second, do all of those actions and experiments that we talked about, then train some sort of model, the details of which we'll talk about in a second, that maps between the initial state, environment, and actions to the outcomes, and then figure out another function that takes all of those things and maps it to desirability, how much we like it. Then we see how well these models work, and if they're good, we stop, and if not, then we go back to the start and keep on going. So some of this process, you actually have to do experiments. That's in, sorry, the colors aren't great here, but that's sort of in this section here. There's some parts that are just statistics, and then some parts that really require you as the human to think about what you're trying to achieve. In terms of numbers, this might look like the following. So here we have some actions which reflect adding species A, B, and C. 0, 0, 0, this means we didn't do anything. 1, 0, 0, 0 means we added A, but nothing else, and all the way down. So let's say we had enough money to do half of these experiments, and then we can observe some of the outcomes here for these top 4 cases. So you can see this is starting to look like a pretty simple machine learning problem. We're trying to predict this from this. It's just a simple function mapping problem, and we'll call that function F. Similarly, for prioritization, we can take our actions and we can predict their desirabilities, and then we can enumerate over the entire action space to figure out which one would have the highest predicted desirability and pick that one as the best action we should have taken. So that's basically solving an argmax problem, if you're familiar with it from the machine learning world. And in this case, you just throw it into the computer and out the answer comes with the right amount of computation. So that's kind of the math of what we're actually doing. It's not that complicated, to be honest. It's taking off-the-shelf algorithms and just reframing them in terms of these sorts of problems. But if you would like another analogy to what's going on here, you can think about an interpretation of this as being interpolation from incomplete data. So we know something about how a community responds to certain kinds of perturbations. From that, we can hopefully learn how it would respond to every other possible perturbation. And I think a nice analogy comes here in the context of art and unfinished paintings. So here's this work done by Da Vinci, which was on view at the Metropolitan actually a couple of years ago. And if you zoom in, you can see that many parts of it are not finished. He didn't get to the faces, he didn't get to the arms, didn't get to the lion. But we don't really need them painted in. We can sort of imagine what they would look like because we know enough about the rest of the painting. We know enough about him. And that's what we're also trying to do in this situation. We're trying to fill in the gaps with limited information. And if we can have sufficiently limited information and still get a good answer, then we're happy because we've basically gotten a shortcut around all of the complexity. We can do a limited amount of work to get a high value result. So what this actually looks like in terms of code, so we can learn that first function just using a random forest algorithm, a little bit of binning to help it out. And then to generalize across environments, if we want to look at more than one environment, maybe cases of climate change, we can just include some environmental variables as additional predictors as well. Then to solve that argmax problem, we actually just use enumeration. It works well enough, but you could probably use some sort of MCMC algorithm as well if you wanted. And then to see how well it works, you can use cross-validation. So it's a very standard workflow. So within this, some questions then begin to arise, like how well does it work compared to anything else? So we've compared this, and I'll show you results in a moment, comparing against a couple other algorithms. First of all, a model-based one, where you fit a Volterra model to data. A residual learning approach, which works pretty well for a lot of machine learning tasks, where you basically fit the model and then try and do some sort of mechanism-free thing on the residuals of that model. We throw in a bunch of parameters into the GLV model and learn on that. And then we try a sequential approach, where we try and select our experiments to do based on which ones we think are going to be the most valuable, that are going to give us the most information about the system, basically the most useful perturbations. Then we also try a really simple approach where we just assume whatever you put in comes out on the other side at some average abundance to make sure that we can do better than a very simple null expectation with an approach like this. And then, of course, the question beyond this sequential approach of what sort of data you should put in, what sort of experiment should you do. So we tried a couple of different approaches here. A mixed one where we just randomly pick experiments. Out of all of the possible experiments, we just pick some and do them. We also try cases where we start with low richness experiments, adding species in just doublets and triplets, which is very similar to what a lot of coexistence studies do. And we also try a dropout approach where we try a lot of high richness additions, which is very similar to what a lot of microbial-based ecology studies will do. And then, as I mentioned, we have this sequential approach as well. So this then, in the next part of the talk, opens up the question of empirically, does this work or not? And which of those choices is going to be the best one for being able to predict and prioritize in communities? And it opens up some questions too, as well, about, how does this work compared to the more mechanistic approaches? How much data do you really need? How should you sample it? Can you generalize? What properties of a data set are useful? And what kinds of prioritizations can you actually achieve? So we put together 10 different empirical data sets. The details don't matter for this talk, but they come from a range of different taxonomic groups, from little microbes up to big trees. They come from single environments, they come from changing environments, they come from low richness systems as low as 5 and as high as 28 species. So we threw together a lot to give a really strong test of this sort of system. And just to illustrate what the data look like and what the machine learning task is, these are the actions in one of our data sets, the soil bacteria. So if you added species number 1, you're orange, and if you didn't, you're white. So you can see there's a lot of combinations of experiments that were done here. And then these are the actual outcomes you get. These are the abundances, relative abundances of the species. I'm sorry, actually absolute abundances of the species and the outcome. So you're basically trying to draw this picture if you only knew this picture. That's what we're trying to do. And then here's another case. This is for a plant case. These are the experiments. These are the outcomes. And we have this here for both dry and wet conditions. And you can see the outcomes are actually quite different in dry and wet conditions, which should be unsurprising from a natural history perspective. And then there's another 8 data sets I'm not showing you just for time and space. So what do we find out? Well, the first thing we find out is that using that random forest algorithm is actually the best way to go. And you can get about 2% to 4% relative error on abundance predictions with less than 100 data points, which is quite surprising in my view, but let me show you this. So here is our scaled error. This is basically percent error on the y-axis. We're log transformed just to make some of the trends clearer. And here you can see the random forest algorithm starts pretty low at under 10% and drops a lot lower very quickly as you increase the number of data points. The naive approach actually does pretty well as well, but we do better than that using this algorithm. The model-based approaches, using anything related to the GLV model, really fall down with error rates an order of magnitude higher. And doing things sequentially, trying to pick cases that are useful, actually isn't much better than just picking things at random. So this is quite surprising. It's telling you that this very off-the-shelf approach, using a simple algorithm and just randomly picking things to do to a community, tells you a lot about your ability to predict. And to reinforce that point about the random sampling, we can now compare for the random forest algorithm what the error rate would look like at the same sample size for different experimental designs, sampling in different ways. And here, what you can see is that I have an animation problem. I'll try and get this at the end if someone has a question. But basically, the inference is that these red dots are always going to be much lower than the other colored dots. You'll have to trust me, but if we delete that white box, I can show it to you. We can also generalize across environments often as well. So in a couple of these data sets, we have multiple environments we can train in, and then other environments we can test in. And here we can also look at how the scaled error varies with the number of training cases. And I do have an animation problem. All right, bear with me for a second. You should always test your presentation first. We'll just delete the boxes. Okay, so hopefully here you can see that in this case, for these bottom 2 data sets, a fruit fly data set and a grassland plant data set, you can see that the error rates go down as the number of training cases increase, indicating that we are learning something about the system. And you can see that whether you're testing in your training cases or in totally new environments you've never seen, the error rate also goes down. Not quite as fast when you're only in new cases, but you can still generalize to new environments. this is telling you, I think, something pretty cool, which is that doing these random experiments in a couple environments lets you predict what's going to happen to a community in a totally different environment. Except if you're this ciliate data case. If you're this data set, it doesn't work at all, which is very interesting. Let's see if I can make this bigger, because I think the rest of the animations are going to get me in just as much trouble. Oh no, I think this is going to work. Okay, back to PowerPoint. So you then might wonder, well, what are the properties of these data sets? Why do some of them work and why do some of them not? So we can make some post hoc models to try and explain how the error rate varies with things like how many species there are, how many species tend to get competitively excluded in the experiments, how many environments we can train on, whether or not the dynamics are deterministic or not. And what we see after doing this is that there aren't really super strong trends when we try and do a post hoc explanation. It's potentially the case that if the data are more deterministic, so if there's less noise, the error rate's a little bit lower. You can see that here. And it's potentially the case that if more species are excluded in the data, the error rate also decreases. But these are pretty weak predictions. So one of the big inferences here is just sometimes it works and sometimes it doesn't. But in general, it works pretty well. So maybe it doesn't matter to explain this additional variation. And then because scaled error is a little bit hard to think about, I also wanted to just show you directly what the observed and predicted abundances will look like for a couple of different cases. You can see for yourself how close we're getting to the right answer for abundances. So these will be plots just for two data sets where we have predicted abundance versus observed abundance. And I have these square root transformed so you can see some of the rare species better. And then these facets here are for the number of training cases. This is if we did 21 experiments. This is if we did 89. This is if we did 264. So what does it look like? Well, if you do 21, you have a lot of noise. Observed and predicted are not that close. If you get up to 89, you've actually got most of the species pretty close to the one-to-one line in black. And by the time you get to 264, nearly everybody is on the one-to-one line. So even without knowing anything about the mechanisms in any of these systems, we're predicting very successfully how these species do in community contexts. And then for the prairie plant data set here, 21, pretty noisy, but we're close-ish to the line. 89, we're doing pretty well. And this data set actually, we only, I think, had about 150 training data points, so I don't have anything to show you further up. But you can see that the trend is going in a good direction. So then what about prioritization? So what sort of things could we successfully prioritize? Could we figure out actions that would get rid of a species we don't like? Cases where the species starts out present and is not present afterwards. What about cases where we want to figure out the communities that have the very highest Shannon's H, so the highest biodiversity? Or what about cases where we just want the total maximum abundance across all of the different individuals in the community? So these are, maybe or maybe not realistic real-world cases, but they're just examples of some tasks you could think of that somebody might like to try and solve. If we could solve them well, we would see that our skill at doing this, as measured by either a true positive rate or a true negative rate, would be high, hopefully close to 1. And the idea here is that, say if you have 16 species and the actions you take are either adding a species or not, there are 2 to the 16 possible actions, which is a really big number. It's about 1 million. So if you were to just randomly try and guess an experiment to do that was going to yield high biodiversity, you would almost certainly guess wrong. So what we're looking at here is how likely are we to find that best one using this algorithm instead, where the baseline expectation is 1 in 1 million if we are talking a 16 species case. If we're talking a 28 species case, your chances of getting it right on the first try is like 1 in 2 to the 28, which is an even bigger number. So how well can we do this? Well, we can actually do it pretty well in most cases. So if we want to figure out actions we could take to get rid of an invasive species, or just any species, our true positive rate is almost always pretty close to 1, and the true negative rates are pretty high. Sometimes we just can't do it at all. You can see there's a couple zeros down here, so sometimes we totally blow it, but most of the time we don't. We do pretty well. For finding high biodiversity, the true positive rate is basically 1. So if we think we have a high biodiversity case, we definitely do. However, our guessing rate, the true negative rate, how likely are we to actually find one of the good cases, starts out pretty low at low numbers of training cases, but as we go up to like 100 cases, we're in about the 50% successful guessing rate. And that's not bad because, as you recall, the null expectation here is like 1 in 1 million, and 1 in 2 is a lot better than 1 in 1 million. And if we go to about 200 cases, you can see we're actually at like three out of 4 cases are going to be good suggestions. And then for abundance, it's pretty much the same story as for Shannon's H. If you can figure out the high diversity, you can also figure out the high abundance. So this is quite impressive. It basically gives us a tool for guessing through all of the complexity of community assembly and finding the ones that are best according to whatever our criterion for best is going to be. So then we could also play the game of asking for post hoc explanations. What are the properties of the data set that might explain why it's more predictable or more prioritizable than others? So maybe it has to do with how many species we have in the data set, or maybe it has to do with the amount of competition, the number of competitive exclusions, like we saw in the prediction case. And when we put in some data here, what we can see is that the true negative rate, our correct guessing rate, does go up as the number of species increases. And there's not really an effect of the competitive dynamics at all. And if we look at this for the other potential prioritization tasks, like making biodiversity high, it's kind of the same story for species richness, and actually the opposite for competition. And then for abundance, kind of a mix between those two stories. So the big takeaway here is sort of similar. It's hard to explain why we sometimes succeed and why we don't. None of these predictors seem to matter very strongly. But given that we tend to succeed quite well and much better than we would expect by random guessing, it's a lot better than nothing. So let's then summarize the main results before moving on to the latter parts of the talk. So first, not many experiments are needed to succeed to do mechanism-free prediction and prioritization. For an arbitrary set of species in these 10 data sets, you can predict any arbitrary community assembly outcome with about 5% error, or prioritize with about 10% error or better with like 100 experiments. That's pretty cool. And the conceptual inference here is that you have this very high dimensional system, a lot of things going on, but you can apparently make this very low dimensional simple representation in your machine learning model and actually get a lot of that complexity out. The second thing is that when you select data, when you pick experiments to do, randomly selecting them is actually a perfectly fine strategy. It seems to be the most efficient use of resources relative to any of the other options. And actually, many of the things that people do in extant studies, like pairwise competition experiments or dropout studies, seem to have much lower benefit. So the suggestion here is that we should just go towards random assembly or something very close to it to gain the most knowledge. And the interpretation here is that when you randomly put things together, you have a lot of cases where you've put something in or taken something out, and there's actually a lot more to learn from than in cases where there's only a few species at play in each experiment. You just need to have the right algorithm to figure out what the linkages are. And then third, it looks like a single set of experiments is best. So we explored this sequential approach where you do an experiment, you train your model, you then say, well, what would be the next best experiment to do? And then you retrain your model. That didn't do any better than the baseline approach, but you can see it takes a lot more time because in each of these sequential cases, you have to wait for the experiment to be over. So you're wasting a lot of time and you don't actually get any more performance. So the big takeaway here is don't do many experiments, do them randomly and do them all at once. And that actually gets you quite a lot of knowledge. And then lastly, this is, I think, the most questionable thing we're not sure about yet. It might be that these mechanism-free models do capture most of the knowledge in the system, and that having additional domain knowledge is not particularly useful. So in this case, we fit this generalized Lotka-Volterra model, which is every population ecologist's favorite model, to all of these data sets. And it did much worse than the mechanism-free approach. And when we tried to learn on top of it using a residual learning approach, we also didn't do any better, suggesting that maybe you can get enough information, or at least all the information in this very limited data, from something that doesn't enforce any ecological constraints. That's a very big claim. I'm not really sure I believe it without trying to push harder on some other systems and models, but it's food for thought, I think, for all of us if it turns out to be true. Okay, so now that's in the last part of the talk, just move on to some of the other ethical and practical considerations. So let's now say we take it for granted that this algorithm does work as well as I've suggested it would, and it might also do in other situations. And maybe somebody who is a better machine learning scientist than me could tune those algorithms or figure out a better one and maybe make it work even better. So what if it did? What would the consequences be? Well, I think one of the first key things that we need to think about are the power and representation issues at play here. So first of all, if we are going full mechanism-free, we're effectively devaluing expert and traditional knowledge. We're saying we don't need it, we don't care about it, we can just roll and accomplish the things we want to accomplish without needing to talk to those people. And that, I think, really changes the power dynamics of who's doing ecology and what sorts of problems they're going to hope to solve and what the consequences on different human communities would be. And that concern is reinforced by the fact that having this domain knowledge, like a model, doesn't really seem to help. So I think that's something that is going to be really critical to explore. And certainly, right, if you have this algorithm, it puts us in this very technocratic vision of doing science where we sort of hand off our thinking to the algorithm and we take the people out of it. I've tried to suggest in the first part of the talk that the people are still necessary to define the problems and to not do the experiments that they think are unethical, but we are certainly tilting in that direction if we go far along this line. And then lastly, of course, if we're going to be applying this in cases like health, access to the technology and to paying for a generation of the data sets might be very costly and then reinforce some inequities currently present in these systems. So I think implementations, you have to be quite careful. And right, we have plenty of technology companies that are trying to get on the right side of this, or at least to look like they're on the right side of it. And if we decide to go forward with this road, I think it will take some very careful introspection about the sorts of partnerships that we would want to build and decisions about who and where this would get applied on before really moving it ahead. The second big issue is the possibility of accidents. So fundamentally what we're doing is we're assembling communities that don't normally get assembled. And when we do that, there could be some unforeseen consequences. First of all, this approach doesn't consider dynamics, but there really are dynamics. And some of the transient stages of these assembly processes may actually move these communities into states that we're not very happy with, and that this approach has nothing to say about. We just know from this what the long-term outcome would be. We don't know how it got there. And it could be that the getting there could be very dangerous. And it could be that if somebody is happy with a relatively high error rate, they may end up accidentally assembling something that has a very surprising and a very dangerous outcome. You just don't know until you try it, and once you've tried it, then you've found out. And this last point here, no insight into what happens if the environment changes substantially. This is something I'd like to talk about a bit more. So I told you before that we can generalize across environments. So how does that reckon with this? Well, if the environment is continuous, we can potentially generalize across it. So if we say train an algorithm at 15 degrees C and at 19 degrees C, I think we could feel relatively confident that we can make a prediction at, say, 23 degrees C, because we have some guidance in our data. But if we have data that are not continuous, if we have, say, the red environment and the blue environment, we don't necessarily know anything about the green environment. So if somebody were to use this in red and then try and use it in green, something very different might happen, and we have no guardrails on that whatsoever in this approach. So that requires some consideration. And here you can see, for example, in Hawaii, this Passiflora species was introduced on purpose with the hopes of some beneficial outcomes, and it actually had this very negative consequence of overtopping quite a lot of forests. And you maybe could have seen it coming if you're a 21st century ecologist in the 19th century, but they didn't then, and everybody has to live with this consequence now. Another key point is with a lot of machine learning approaches, they're very energy intensive. You've probably seen a lot of that in the news, especially with these large language models. That is, at least for love, fortunately not a concern. So what we've done so far, all is just like off-the-shelf stuff that runs on a laptop. All the results I showed you, I think run in about 30 minutes on a cluster. And probably use maybe 10 kilowatt hours total. So it's orders of magnitude energy lower than some of these other big applications. And I think whatever the ethical considerations are here, they are not around energy use and the consequences of higher energy use. So we can sleep easy there. The biggest thing that I'm actually worried about that I think is worth discussing is the possibility of adversarial use. So we've framed all of these desirable outcomes as things that we want, but we could also imagine things that we don't want, things that could hurt somebody on purpose. So biological warfare applications. So you could imagine maybe intentionally causing the extinction of a species, or reducing biodiversity, or promoting the establishment of a disease. I personally have not yet tried to use this framework to accomplish any of these, but it would take 2 lines of code to make us do the opposite of what we've done so far. And I think it's not an unrealistic fear. There was a recent very interesting study in a Nature journal where they took one of these algorithms that's used to discover drugs for cancer treatments, and instead of doing that, they asked it to find drugs, or rather molecules, that are more toxic than known toxic chemicals. So they gave it VX, the nerve agent, as one of their targets. And through the use of this algorithm, they discovered 2,000 compounds, never synthesized before by humans. And of those, about a third of them were at least as lethal or as up to 5 times more lethal than this known nerve agent. They, of course, didn't publish the structures of these, but they could have, or they could have just gone and synthesized them if they wanted. So it's, I think, a reasonable possibility for our future. And with this sort of technology, if it turns out to actually work quite well, I think we have to be thinking quite substantially about regulation and other approaches to make sure that the data and the applications stay on the positive side. Okay, so I hope that gives you some food for thought on the technology, even if we're not quite there yet. So where does that leave us now at the very end, looking forward? So I think there's 4 sort of key points for you to leave this talk with. The first is that we're starting to move beyond mechanism only thinking in community ecology. I'm certainly not the first one going there, but I think there's a wave going there now. And you can see there's a lot of different issues that come up and that are at stake when we go in this direction. And I imagine we'll see more things in this vein soon. Second, I think previously when we don't know the mechanism and we don't have the data, we've generally gone for the give up option. But I think there may be some more opportunities for progress than we've previously thought were possible, and this is maybe just the first toe in that water. And then third, it seems to be the case that if it did work, there's a lot of impactful prioritization applications across different ecological domains, and I highlighted 4 or 5 cases for you at the front of the talk. So we may be able to really make some progress there in ways that we haven't tried before. And then lastly, I don't think we're ready for any of those applications yet. I think we have to do a lot more work on the theory and validations and other experimental cases to really make sure that we're happy with how this works before we go and try anything in the real world. So if you're interested in that, I'm very happy to talk about that later on. So I will, I think, end the talk there. A lot of people have given input and suggestions. And if you do want to try this yourself, all of those data sets and all of the code, it all just runs. It's all on GitHub. You're welcome to try it. Feedback is welcome. Thanks, everybody, for your time.
Thank you, Dr. Blonder. We have about 15 minutes for questions. If anybody wants to start, I will take a peek at people online too.
Barbara, do you want me to pick the questions, or are you going to do that? Okay. Yeah, let's start up here.
Thanks. This is really interesting, and I have lots of questions I can't wait to ask people when it comes out. But in the meantime, can you explain a little bit more about the temporal assumptions in the outcome?
Mm-hmm.
In the predictions, is the assumption that the answer you get is in the same time frame of the experiments that were done, or how are you interpreting that?
Yeah, so the question was, how do we think about the timescales? Is the timescale of the outcome the timescale of the experiment, or is it clear at all, and how do we think about it? So the answer is it could be anything. You as the scientist would get to decide that. I would imagine most people would be thinking about an outcome in the equilibrium sense or in the quasi-equilibrium sense. So you do something and you wait for whatever transient dynamics there are to sort of stabilize out, and then you measure the outcome. But you don't necessarily have to. You could pick any arbitrary time point and the algorithm won't know any better. You can see there that there are some risks as well. If you pick the wrong time point, you may end up predicting something that doesn't actually stick around longer in time. On the other hand, maybe you're actually very interested in what those transients are and you would like to see, or maybe the system fundamentally doesn't have an equilibrium, it's just bouncing around, but you really still do just want to know what happens after 3 months or 5 years or whatever else. So time is implicit in this approach, and time is on purpose collapsed out. I think the reason this works relatively well is that it sacrifices all of that dynamical understanding for being able to do the outcomes. It's basically taken all of the information in the data and just gone to try and predict the outcome rather than trying to learn anything about the rules or the intervening situations. So you gain something, but you also lose something.
Can I ask just a follow-up, because it leads to my second question?
Mhm.
So all of your examples were kind of single taxonomic level. In the interest of kind of community function, do you think you could use this same method to predict multiple levels of that community?
So the question was, could we do this across multiple trophic levels or multiple other biological levels? All of the examples have been at one level. I don't know because we haven't tried, but in principle, I don't see why the math wouldn't work out just as well. It would just reflect adding a couple more columns to the data set. And the algorithm doesn't know that these species are at the same trophic level. It just knows that we've measured their abundances. So I think it should work just as well across trophic levels. That being said, the data sets that I was able to find in the literature all just happen to be at a single trophic level. And if anybody in this room knows about any combinatorial community assembly experiments where there are things like predators or other things, I would love to see those and we could put them in and get an answer to that. Yeah, in the corner.
Thank you for your talk. I was wondering I was wondering in your model of Rotella, how are you accounting for non-pairwise interaction between species?
Mm-hmm.
And if you're not accounting for that, how that will impact like the success of your results?
Mm-hmm. So the question was, are we accounting for more than pairwise interactions between species and how would that influence the results? So right now, the best performing algorithm was the random forest algorithm. And in that approach, the variables are put together in decision trees. And I think we constrained our maximum tree depth to be 10 in this current implementation, meaning that in principle, you could have a 10-way species interaction being included. So it's very flexible. The interactions are probably pairwise because that's the first level of branching in the tree, and it may have pruned some of the trees there. But it could go to much deeper interactions and other sorts of non-pairwise interactions. And I think that's the value of this, because instead of assuming a parametric model form where we only allow pairwise or maybe three-way interactions, we're very flexible. We don't know, we don't care, it could be anything. And I think that's why we're able to predict better than some of those other approaches. Yeah, in the back.
Really cool talk. One of the things that struck me about midway through, you were showing some plots of like predicted versus observed outcomes, which as you increase your sample size, your predictions got better and better. Even at those high sample sizes, you end up, I think, if I read it correctly, with cases where the predicted abundances are 0, and you get this whole big stack of observed abundances from 0 to a lot. Yeah, these ones. So along the left side of those figures, what's going on there? Why does the model not capture those? Do you have any insight into what that means?
Mm-hmm. Yeah, so the question is, how come we get some of these weird zeros? How come we're really not doing well predicting in certain cases? I think the reason for this, so you can see here, these are cases where we predict 0, but the actual abundance is higher. In data sets like this for community assembly, we often have a lot of zeros in the actual data, cases where species are not found. And for a statistical approach like this, that creates a zero inflation problem. The model ends up biased towards often predicting 0 when it shouldn't. So I think what's going on here is just simply the model having trouble with too many zeros in the training data. We largely got around that by, instead of predicting abundance as a continuous variable, we binned it as a categorical variable. So we had a 0 bin, and then we had like a small and a medium and a high. Actually, I think 10 categories, but the key thing was that there is explicitly a 0 bin. So we were able to get rid of a lot of our false zeros that way. But you can see we're still ending up with a lot of false zeros even still. So that's the primary way things fall down. I think ultimately, a model that explicitly incorporates a hurdle process would be a better thing than what we currently have. But finding off the shelf in R something like a random forest, but with a hurdle that accounts for multivariate data and correlations in those variables, didn't find anything. And because that's not my core area of expertise, we just went with something that would work. So I think there's actually quite a lot of ceiling in this approach to have better algorithms under the hood within this conceptual umbrella. Yeah.
So one of the obvious applications of this would be trying to predict disassembly or
So the question is, for cases of rapid climate change, could we make this work? And the challenge is being maybe not being at an equilibrium state, and then also not having random data to work with. Am I understanding it right?
Better said than I do.
Okay. I've thought about that a lot, and I'm not sure I have a great answer to these sorts of questions. The primary reason I'm a little bit concerned is that you do have to do the experiments, and the experiments have to take some amount of time to run for you to be able to observe an outcome at a level for which you get an interesting answer. So if you're thinking about, say, what's the 30-year impact of climate change, in this approach, you would have to wait 30 years to train the model, at which point it would no longer be interesting to have the answer. So I think for those sorts of questions, this is probably the wrong approach to use. That being said, if you're less concerned about the time, but more about the underlying environmental variable, you could, say, do a bunch of 2-year experiments that push the environment to what the 30-year environment would be in the natural case. And in that version, if you believe in the ability of this algorithm to extrapolate, you could then say, my 2-year high push experiment is equal to a 30-year low push experiment. And then I think you might have some success. But probably I think the real applications for this are going to be in shorter timescale applications, like some of the revegetation, invasive species, and synthetic ecology, microbial ecology areas. We'll see. Those are also scales at which doing the experiments is a little bit easier, thinking about the challenges of, as you say, randomly assembling forest communities.
I'm just going to follow up on Josh's question and ask whether you've thought about taking advantage of the fact that climate change impacts are happening faster in some places of the world than others, and using that as sort of a substitution, like a natural experiment, treating them as experiments when they're not actually, to get at some of the forecasting.
Yeah, I think that's super interesting to think about cases where we can look at observational data and treat them as experiments. In this study, we only use true experimental data, and I would love to do a follow-up where we try and use some natural experiments and see how well this works there. And I just simply don't know the answer, but I think it's a great idea. And again, if anybody has already collected that sort of data for other purposes or wants to work together, I would love to talk to you about it.
I have a similar follow-up question about biotic interactions that we talk a lot about no analog futures and communities that will exist that we can't imagine existing for reasons that we don't know yet. Some of the more interesting things that I think about a little bit but haven't actually done anything to explore are these totally new biotic interactions that don't have experiments because we haven't imagined it. So trying to kind of capture these outcomes where the outcomes are only predictable if you understand the mechanistic interactions between the species. Have you thought at all about love in the context of those types of biotic interactions that are sort of unknown? And
Yeah, so I think that's super interesting. And I think the love answer to that would be go do an experiment and put some of those together and see what happens. And from that, maybe you can extrapolate to other novel cases that you haven't yet seen. So I think fundamentally without training data, you don't get anywhere. But if you're willing and able to get some training data, I think a lot of the no analog space suddenly becomes very openable, and we possibly get a lot of insight into what's going on. It just takes, if you sort of imagine this high dimensional predictor space, you just need a couple of anchor points somewhere in that space. And from that, I think this low dimensional machine learning representation can actually predict out the rest of it fairly well based on what we've seen so far. So it could be cool. The question is it ethical to go do that? And that depends on the system.
I have one more question, if you don't mind. So riffing on that a little bit, I was thinking about the desirability scores. And, if you bear with me, like the future where AI makes decisions about what action to take based on some maximization of, optimizing on some score. You can imagine cases where even in community ecology and restoration where you're trying to optimize for two different things that seem like they're opposed to each other. And there are not just two, but usually multiple things that you have to consider. Have you thought about how to transfer that kind of a problem where you're trying to have different desirability scores for different outcomes into the love context where, you're doing the same experiment and you could have multiple different desirability scores? Is there a way to optimize in a multidimensional desirability output space?
Yeah, so how do you do a multidimensional optimization there? I think I have a couple answers. The first would be you could, of course, take those different dimensions and weight them in some way to come up with a single variable and then just do a single variable optimization on top of that. So you could say, like, I'm willing to pay a 30% penalty for X in order to get a higher value of Y. So I think that would be the simplest way to build it in. And of course, the desirability scores, once you have them along any axis, then the rest of the workflow is just computational. So you could play that game multiple times and just see how it comes out. But I think my real answer, and how I would really imagine this being used, would be more that we might try and apply it along each of those axes independently. And instead of asking for just the one single best, we would say, give us 100 possible options. And then we would take those 100 options and play the same game for the other axis. And we'd have 200 options. And then we would take those to some real people. And we would say, could you look at these? And which of these would you be happy with? Which of these are you worried about? Which of these is a total non-starter? And then the actual action we would take would be decoupled from the machine. It would still have that extra layer of human oversight. And I hope that's the way a lot of these decisions will go in the longer run.
I have lots of follow-up questions that I will ask you over lunch. You guys are all welcome to join us. Please join me in thanking Dr. Blonder.